Sequences of DNA that encode particular proteins have different forms, because its sequence can vary from individual to individual. Genes. DNA consists of the building blocks A, G, C, T. Adenine, guanine, cytosine, and thymine. A part of a DNA molecule that contains the instructions for constructing one specific protein. Gene. Humans have about how many genes? 35,000. All the genes in a human cell make up our what? Human genome. The study of inheritance of characteristics. Genetics. Consists of a tightly wound DNA molecule in proteins. Chromosome. Humans have how many chromosomes? How many comes from the mother? How many comes from the father? (46, 23, 23). A chart that displays chromosomes in size order. Karyotype. A normal karyotype has how many pairs of autosomes aligned in size order, plus the sex chromosome? (22). The process whereby cells undergo spermatogenesis or oogenesis produces sex cells with half the number of chromosomes found in other cells of the body. Meiosis. The patterns in which genes are transmitted in families are termed blank blank blank. Modes of inheritance. An educator, monk, and gardener named Blanc Blanc deciphered the modes of inheritance in the 1860s. By breeding pea plants and following easily visible traits over several generations. Gregor Mendal. Because a gene consists of hundreds of nucleotide building blocks, it can exist in variant forms, called blank, that differ in sequence. Alleles. The combination of gene variants in a person's genome constitutes the blank. Genotype. The appearance or health condition of the individual that develops as a result of the ways the genes are expressed is the blank. Phenotype. Disease-causing alleles are blank. Mutant. An allele whose action masks that of another allele is termed what? Dominant. The allele whose expression is masked is blank. Recessive. Carried on a non-sex chromosome. Autosomal. Carried on the X chromosome. X-linked. Carried on the Y chromosome. Y-linked. The chromosomes have already duplicated, they coil and become shorter and thicker and visible under the light microscope. The duplicated homologous chromosomes pair, and crossing over occurs. Prophase I. The centrioles are at opposite poles of the cell. The pairs of homologous chromosomes become arranged on a plane equidistant from the poles called the metaphase plate. Metaphase I begins when the two chromosomes of each bivalent separate and start moving toward opposite poles of the cell. As a result of the action of the spindle, the sister chromatids remain attached at their centromeres and move together toward the poles. A key difference between mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis I, whereas in mitosis they separate. Anaphase I. Now a haploid set of chromosomes is at each pole, with each chromosome still having two chromatids. A nuclear envelope forms around each chromosome set, the spindle disappears, and cytokinesis follows. Telephase I. While chromosome duplication took place prior to meiosis I, no new chromosome replication occurs. Before meiosis II, the centrioles duplicate. This occurs by separation of the two members of the pair, and then the formation of a daughter centriole perpendicular to each original centriole. The two pairs of centrioles separate into two centrosomes, the nuclear envelope breaks down, and the spindle apparatus forms. Prophase II. Single chromosomes align on the metaphase plate, much as chromosomes do in mitosis. This is in contrast to metaphase I, in which homologous pairs of chromosomes align on the metaphase plate. Metaphase II. The centromeres separate, and the two chromatids of each chromosome move to opposite poles on the spindle. The separated chromatids are now called chromosomes in their own right. Anaphase II. A nuclear envelope forms around each set of chromosomes. Cytokinesis takes place, producing four daughter cells, each with a haploid set of chromosomes. Because of crossing over, some chromosomes are seen to have recombined segments of the original parental chromosomes. Telephase II. Blank million combinations of 23 chromosomes from each male and female produces the possibility of more than blank trillion genetically distinct individuals. 870. Blank during prophase I contributes even more variability. Crossing over. Every characteristic requires two blank in order to be expressed. homologous genes. Chromosome charts are called karyotypes and are used for diagnosing blank blank genetic abnormalities.